you.
Hello everyone, and welcome to today's TSU Technique Talk. Technique Talks, a series of educational courses, answer the question, how can I get this experiment to work? Course instructors teach about how to get a new method or technology set up in the lab, common challenges or pitfalls to avoid, and troubleshooting steps that will help scientists achieve publication-worthy results. I'm Nikki Spahich, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our instructor, Dr. Craig Powell, will explore approaches for optimizing your IHC and IF staining workflows. I'd like to take a moment to thank our webinar sponsor, Vector Laboratories. They've provided us with some helpful resources related to today's course, and you can download them directly from the Handouts tab located on the right side of your screen. This course is meant to be interactive. Please send us your questions or comments at any point during the discussion, and the instructor will address as many as possible during the live Q&A. The Technique Talk will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our speaker. Dr. Craig Powell is the Director of Technical Services at Vector Laboratories. Prior to joining Vector Labs, Powell obtained a PhD in physiology from the University of Queensland, Brisbane, Australia, where he first learned histological techniques while studying reproductive biology in bats. Subsequently, Powell conducted his postdoctoral research at the California Primate Research Center at UC Davis, where he examined cellular migration and differentiation using tissue-based staining methods in early post-implantation primate embryos. In his role at Vector Labs, he leverages his deep technical expertise to help researchers optimize and troubleshoot their experiments. Let's just make sure the slides are up and running. Okay, Dr. Powell, take it away.
All right, terrific. Thanks very much for the introduction, Nikki. It's certainly a pleasure to be speaking here today, and hello to everyone that's joined us. The applications of enzyme-based immunohistochemistry and standard epifluorescent immunodetection that we're going to be discussing today are not particularly new or novel. However, both of these techniques are widely used across the life sciences disciplines, everywhere from neurobiology, cell biology, immunology, histopathology, to help elucidate protein expression in cell and tissue specimens. And despite these techniques being well established and certainly widely adopted, as I mentioned, they're not infallible. And when working with biological specimens, unusual specimens, the outcome is certainly far from guaranteed. So what we'll be looking at today are some of the commonly encountered issues that you might encounter or find when running your immunohistochemistry or immunofluorescent application. We'll first look at our background or non-specific staining. What is it I'm referring to when I talk about background, sources of background, and things that you can implement into your workflows to help overcome or eliminate the background. Then we'll toggle to look at an absence of staining, which can be quite confounding to some investigators. But what it does do is allow me then to segue into talking about the appropriate control or implementation, I should say, of positive and negative controls in your applications. Then we'll finish up the webinar with a brief discussion on inconsistent staining that does plague some investigators. All right, so let's dive into it right now. So what is it I'm referring to when I talk about background? And I've got a little bit of a definition here: non-specific staining or signal observed on the test specimen. And I am using the phrases background and non-specific staining here interchangeably. But really what I'm getting at is it's unwanted staining on your test specimen. And it might be observed as a trace or a weak staining, maybe an overall toning across the tissue section. That might not be too problematic, particularly if your specific signal is highly expressed and you've got a nice staining for that, and you've got a very good demarcation between the signal to noise ratio. However, things tend to compound a little bit more when you start to see more moderate staining, maybe more widespread across different tissue types, and certainly then when you get severe staining, almost like a blanket across your section, for example, or cell prep. I'd say if you are getting severe staining, there's probably something fundamentally wrong with that assay that needs to be addressed. So more than likely, most investigators would probably encounter trace or moderate staining. But regardless, the background can certainly arise from one or more sources. Now, as I've indicated here, it could be inherent or endogenous to the particular specimen that you're working on. Certainly could arise from the detection reagents, or indeed a combination of both the specimen and the reagents that you've chosen to use. So just to clarify a little bit for investigators that might be new to the field and are listening in, what is it I'm referring to about background in IHC or immunohistochemistry? It's the oxidized substrate reaction product that's precipitated out or deposited on that particular specimen, whether it be a section, for example, or cell prep. And the example would be, say, a brown DAB staining, diaminobenzidine staining. For immunofluorescence, it would be a visible interfering fluorescence in the same spectral wavelength as the specific detection fluorophore. So the example would be if you're using a fluorescein, a green conjugated secondary antibody to look at a target antigen, and you've got some confounding fluorescence in that same green spectra. Okay, very quickly, some of the issues or problems that arise from the background, certainly interference with the target antigen that you're looking for, certainly referred to as your specific staining. And the issue there is that you can identify then or falsely identify staining. It's a false positive staining, and you may go off on a tangent there thinking that is correct staining when in fact it's incorrect, it's false data. And background certainly calls into question the validation of your assay, and you start scratching your head about what is specific, and certainly throws into question the credibility of what you're reporting on. And the goal, whether it be immunohistochemistry, immunofluorescence, is to obtain a clear, unambiguous, unequivocal view of the target antigen or antigens that you're looking for. And certainly with any troubleshooting endeavor, it is a two-step process. And certainly for immunohistochemistry and immunofluorescence, it's no different. Certainly you have to identify the source or the sources of the background that you're encountering, and then put in place or implement specific methodologies and strategies to overcome that background staining. We'll be discussing some of those going forward. But let me just digress here for a moment. Sometimes on technical service, in talking with investigators, they might be saying, I've had some great staining the last couple of weeks. It's been very clean and haven't experienced any background problems. But this week, I've got background and I'm not sure what I've done wrong. Nothing has changed. I'm using the same antibody, the same block of tissue, the same detection reagents, and what am I doing wrong? And it's not that you're doing anything wrong, but more than likely in these instances, you might be working with something like a heterogeneous tissue block. So indeed, something has changed at your end that you might be working with. And what I've got this example as you're cutting into a block and you're cutting off these sections in a nice ribbon here, you might be getting further into a tissue block where there may be different cell types, for example, different cell components, different tissue components, maybe more Fc receptors, charged particles, macromolecules that could be contributing to background. There could be varying levels of antigen expression as you cut into a block, and indeed vascularization, whether it be angiogenesis, neovascularization, whatever you want to refer to that as, but really introducing more erythrocytes, for example, that may have more peroxidase activity associated with it. Just to give you an example of that, whether it be a xenograft model or tumor model, something like that. Inflammation, maybe is an influx of more leukocytes there, whatever they might be, mast cells, whatever, T cells, B cells. that could be contributing to background. And certainly depending on the specimen you're working on, there's a potential for necrosis to be present. So one or more of these components, as you're cutting into a tissue block, may be contributing to background. So just be aware of that and flexible in your approach when you're doing these particular applications. All right, so let's have a quick look at the immunohistochemistry workflow. Basically a series of sequential incubation steps I've highlighted here in this block of text. So just bear with me, probably the most boring slide I've got here in my presentation. But nonetheless, I think it's important just to have a look at what we've got here. So step number one, tissue prep. What I mean by that would be potentially fixation, cutting of the sections. This may or may not be under your given control, depending on the setup where you might be in a CRO organization. You might just be handed a block of tissue and told to work a miracle on it and generate staining. So I understand that some of those things might not be under your control. But nonetheless, something like antigen retrieval probably is. And it's associated mostly with, I think, a sensitivity and certainly reproducibility of the stain. Another word for reproducibility is consistency. And as I indicated at the start of the webinar, we're going to be discussing a little bit about that later on. So the main focus for troubleshooting we're looking at here are steps 2 through 5 that would certainly be under your control when you are doing the immunohistochemistry assay. So just keep a look at steps 3, 4, and 5 here, and we're going to make this into a little bit of a diagram on this next slide. Okay, a little bit of a pictorial interpretation of what we just had. Your primary antibody incubation here, your secondary, in this case, enzyme polymer conjugate, and then you're reacting with a substrate. So, for example, this would assume that you've already done your blocking and quenching. Here's your, for example, mouse primary antibody. You do your buffer washes, then you put on, say, an anti-mouse peroxidase conjugated secondary detection reagent, and then react or develop that with a DAB, for example, substrate appropriate for that enzyme. So the idea here, we're going to be working from this particular graphic. The idea to start troubleshooting is to run defined deletion controls. Now, they're a special type of negative control. So just keep this in mind as we go through the procedure. And the thing you want to do is to eliminate certain steps of the procedure to help pinpoint the source of the background. So number one, you would omit the primary antibody, you would omit the secondary detection reagent, and simply just apply the enzyme substrate. So this is what we're going to be looking at as the first deletion control. So there was a little bit of PowerPoint magic there. We've got just applying the enzyme substrate onto your tissue preparation. And if you do see any staining with this particular deletion control, it could be due to one of these factors that I've indicated here. It may have been inadvertently left out or omitted from the workflow. Potentially, it may be further diluted than what is required for the amount of enzyme that's present, or indeed it might be past its expiration date, or indeed there is a time factor there. Maybe it needs to be applied for a longer period, for example. This might be in cases where you might have increased vascularization, as I mentioned on that other heterogeneous tissue block slide. Certainly, just keep in mind also, depending on the enzyme-based system you're working for, make sure you're quenching for the appropriate enzyme activity. So if you are using a peroxidase-based detection methodology, make sure you're quenching for peroxidase activity, not alkaline phosphatase. Okay, some commonly used methods here for quenching endogenous peroxidase activity. Now, there might be some listeners out there who might be tuning out and saying, oh, we've already got this established in the SOP. You know, we do this already. I challenge you on that and say, is it being applied effectively and appropriately? And when the question of background comes up, or you encounter background, interrogate each part of the workflow that you've got, and just simply don't blow it off as like, oh, we've always done it that way. And so I come back to the points of, is it done appropriately and effectively? And I'll use an example. There are some investigators who might be using, say, frozen material that's highly vascularized and using this 3% H2O2 in water, and they're getting a lot of bubbling on that preparation. And they're going, oh, this is great. It's quenching all those heme groups. I'm not going to get any background from peroxidase activity. But unfortunately, they may not be aware of the fact that what that is doing is really destroying the underlying tissue architecture and morphology. And there would be a better choice in that given application whereby you'd go to more of a methanol-based use of this H2O2. So again, I just challenge you a little bit to consider these things, and don't be a victim of lab dogma like, oh, we've always done it that way. There may be a better methodology. And indeed, for those investigators that may be struggling with prominent or residual peroxidase activity, there are other choices. I've highlighted a couple of references here, and certainly there might be commercial products that you want to investigate as well. For those investigators who are looking to use an alkaline phosphatase-based detection methodology and unclear about what would be suitable there, the use of levamisole up to around about a 5 millimolar concentration applied directly into the alkaline phosphatase substrate working solution is usually the way that this is addressed. Now, just note that two things. One, levamisole is a reversible inhibitor of alkaline phosphatase, and as a result, it is put in at a different part of the workflow than when you would quench for endogenous peroxidase activity. So it's actually put in at the end with the enzyme substrate working solution. I know it sounds a little counterintuitive, but that's the way that particular product does work, or chemical does work. There are other applications. I've highlighted a publication here using acidic solution, and then certainly commercial products may also be utilized for this. Okay, so we've just worked through a first deletion control, and we're revisiting this schematic or cartoon that I put in here. And for the first deletion control, we simply applied just this end part, the substrate itself, to see if there was any color development. So if you work through that, maybe eliminated that or didn't find any staining, let's move on to this second deletion control, which omits the primary antibody. It does, of course, put on your quenching and general blocking, but omits the primary, and then you put on this secondary detection reagent and the enzyme substrate. Again, focusing a little bit just on immunohistochemistry for the time being. What this does test for is whether or not that secondary detection reagent is the source or of background or binding to components in the tissue. And I've got a little table down here. Indeed, if you do see staining occurring with this second deletion control, more than likely it's due to something such as species cross-reactivity, inadequate or indeed inappropriate blocking, and maybe inadequate buffer washes, not doing enough or indeed too short buffer washes. There are some other considerations too, such as concentration, but I think that a lot of these products these days are offered as pre-diluted, ready-to-use reagents, so I don't think that is particularly an issue. And then, standard lab temperatures, for example, I don't think factor in either, and there's usually established protocols about the time. So let's take a little bit of a deeper dive here into species cross-reactivity. What is it that I mean by that? An example, species on species, would be something like applying a mouse primary antibody onto mouse tissue sections. And usually this would be with an unconjugated mouse primary antibody. And the idea then would be you follow through or detect, as we had in our schematic there, using a secondary detection reagent, an anti-mouse conjugate, whatever that might be. And this is a little bit where some of the fluorescence does come in. It could be an anti-mouse fluorophore or anti-mouse enzyme conjugate. If that is the case, that secondary antibody in this application, is not smart enough, or use that, to differentiate between the mouse primary antibody immunoglobulin and immunoglobulin that's present in that given preparation. So it will bind to all inherent endogenous mouse IgG in addition to your primary antibody. If that is the case, certainly there would be, or there are, some commercial blocking options, as I've indicated here on these pictures, that are available out there. If you are applying, say, rabbit on rabbit or goat on goat, I don't know about commercial entity or products available for those. But if you are facing something like that, certainly there might be options to look into changing the primary antibody species. Let's go back to the example here of mouse tissue. If you are, faced with this scenario of applying a mouse primary on mouse tissue, can you change it to maybe a rabbit or a goat, and then you're applying an anti-rabbit or anti-goat that has a significantly lower or not even any cross-reactivity with mouse IgG? That would certainly be an approach. In some cases, though, that is not feasible or not practical. Therefore, you could look at conjugating that primary antibody directly, either possibly with a fluorophore, if your methodology is immunofluorescence, or maybe with an enzyme directly for immunohistochemistry, or indeed, to make it a little more flexible, you could biotinylate it and then come in with your choice of streptavidin fluorophore or streptavidin enzyme conjugate. Certainly, staining with this second deletion control also arises when using closely related species. The example I will use, let's just say, again, let's go back to mouse tissue, very commonly used species out there in many research labs. Applying a RAT, that's R-A-T, primary antibody onto mouse tissue and detecting with an anti-RAT secondary antibody. There is a certain degree of species cross-reactivity between rat and mouse, relatively closely related rodent species. And you're going to get some background if you just use a standard anti-rat secondary antibody. So look for alternatives such as pre-absorbed reagents, an anti-rat mouse-absorbed secondary antibody. What this means is it's a secondary antibody, anti-rat, that's been run down a column of mouse IgG. Anything that recognizes mouse has been physically removed. The run-through has been collected, and that's the anti-rat mouse-absorbed product to help eliminate or significantly reduce cross-reactivity to those tissue elements. Another little trick you can do, actually, is if the cross-reactivity between that secondary antibody and the tissue species is low, let's just say around 3 to 6%. Let's just use an example here of, again, that mouse tissue, but maybe applying a guinea pig primary antibody, and you're going to put on an anti-guinea pig secondary antibody, but it's got, let's just say, a 6% cross-reactivity with mouse IgG. What you can do is dilute that secondary antibody in a small percentage of mouse serum, let's just say a 2 to 5% mouse serum in buffer. Let that sit for, say, 30 to 60 minutes at room temperature on the bench, and then apply it to the primary antibody. And that should significantly cut down on interaction with the tissue. Okay, some other sources of staining with this secondary deletion control. Again, we've omitted the primary, putting on that secondary and the enzyme substrate, for example. Look at the serum you've got. The idea of this serum is to block, as I mentioned earlier, Fc receptors, macromolecules, charged particles, cheap source of high protein. Use serum from the same species in which that secondary antibody is raised. So, for example, if you're using goat anti-mouse, use goat serum. Horse anti-mouse, use horse serum. Just make sure you've got the correct dilution and put it on for an appropriate period of time as well. On a tissue section of, say, 10 microns thickness or less, usually 30 minutes at room temperature is adequate. If serum is problematic for whatever reason, or you're juggling a lot of different sera, there are alternatives out there. Indeed, plant-based animal-free reagents that actually are fairly useful, and they do double up as universal blocks and diluents to serum. So that might be a cost savings for you in the long run as well. Use these as universal blocks and diluents. And in some other cases, mostly with thicker tissue preps and maybe some other preps like frozen sections, you may want to consider using a detergent like a Tween 20 or a Triton X100 up to about 0.5%. Certainly useful for permeabilization. However, my point here is certainly cut down on some potential hydrophobic interactions as well. Okay, just following on here again, staining with this second deletion control. Potentially, you may see an overall toning in your sections, for example, with enzyme-based polymers. If you skimp on the buffer washes, if you don't use enough or don't run them as long as perhaps you could. So again, throw in an extra wash, run it a little longer. It might clean up the background. Hopefully you can see that there's no given toning in this particular adjacent tissue section here. So just be aware of that. Some of the other contributing factors I mentioned, probably not going to factor in largely in most of these issues associated with a staining in a second deletion control, but just be aware of them anyway. Just very quickly, for investigators that might be using, say, a two-step methodology, whether that be a two-step enzyme-based polymer methodology or indeed an avidin biotin-based methodology, you will have to slot in an extra deletion control just to make sure that that other component, that second step, is not contributing to background in your procedure. And I've got here just a couple of suggestions if you are using an avidin biotin-based detection methodology with a biotinylated secondary and a streptavidin conjugate. Just see whether or not there might be any binding to inherent or endogenous biotin activity in your tissue prep, and if so, incorporate some of these additional blocking steps. Okay, so now you've worked through, or we have worked through, the first deletion control, the second deletion control. Let's throw the primary antibody into the mix again and see what we get. And if you do see an appropriate staining, have a look at that primary antibody and some of the parameters associated with that. More than likely, most people or end users that are listening in here probably get their antibodies from a commercial source. It might be validated, or if not, you may want to search for a more validated primary antibody, maybe a more purified format as well. But just keep an eye on things such as the recommended titer or dilution on that, and the diluent as well. I've had investigators run into problems with an incorrect diluent when using some of these antibodies. Some investigators out there listening might be in a discovery lab or working with other folks generating their own antibodies. Maybe the antibody itself is from a non-purified source, possibly from one of these particular things such as an ascites or culture media, for example, indeed even whole sera. If that's the case, you want to do a couple of different things. Number one, you're obviously going to have to run a titer series to look for that signal to noise ratio. Where's the most optimal dilution to run that primary antibody? In addition, I'd highly recommend diluting it in a blocking agent, appropriate serum, same serum that you're going to use for the species that secondary antibody is raised in, and throw in some detergent as well. Again, just a small amount, about 0.1%, Tween 20, Triton X100. I prefer Tween 20 actually. And again, just hopefully cut down on some of that stickiness that you might get with some of these non or unpurified antibodies, or particularly polyclonals you might find. Another quick little trick you can use is, as we mentioned in a prior slide, diluting that antibody, this unpurified primary antibody, in buffer containing a small amount of normal serum derived from the same species as the tissue. Again, we can say if you're using a mouse tissue, use 2 to 5% normal mouse serum to mop up some sticky stuff in that unpurified prep, leave it for about 30 to 60 minutes on the bench top, just at room temp, and then apply it to the tissue. And that should hopefully clean up some of the stickiness if it's derived from ascites or culture media, for example, things like that have other proteins in there that may be a little sticky towards some of the tissue components. Okay, let's pivot now over to immunofluorescence. Certainly background is something that also does happen in immunofluorescence, and it may be arising much the same from immunohistochemistry, from the detection reagents. It might be endogenous to the tissue, or a combination of both. Let's revisit this particular workflow we looked at for immunohistochemistry. It's a little different from immunofluorescence, obviously using not enzyme-based reagents, but a fluorophore. But certainly it presents the same sort of approach to do these defined negative controls. So, for example, if you've simply got a primary antibody followed by a fluorescent secondary, drop out this primary antibody, just put on the secondary detection reagent, see if there's any species cross-reactivity, for example. Have a look at the protein blocking. Is it appropriate for your given application and detection reagents? Has it been left on for the correct amount of time? And run those particular defined negative controls as you would, as we described here, for immunohistochemistry in the prior slides, and employ the similar tricks that we mentioned. But I will say that one thing that investigators using immunofluorescence encounter that you don't using enzyme-based immunohistochemistry is tissue autofluorescence. And this can be a bit of a bugbear, I'll use that phrase, for some investigators. It's fluorescence that is inherent or endogenous to their given preparation, that might be arising from a number of different cell types, or indeed tissue factors, collagens, elastins. Red blood cells are particularly notorious, or indeed due to things such as an aldehyde-based fixative. It can be induced through the use of a formalin, paraformaldehyde, or even a glutaraldehyde, for example. And you can imagine on this given slide here, in this green spectra, if you did have a fluorescein or a 488 conjugate, it really interferes with the signal you're looking for. But the confounding thing, of course, is if you think, I'm just going to go to a red fluorophore, well, unfortunately, tissue autofluorescence just doesn't stop at the green spectra. It's across the spectra, even into the far red. So there has to be, some sort of methodology of quenching or blocking this tissue autofluorescence. Now, what I've got here then on this particular slide is a series of essentially adjacent sections of human pancreas with various treatments. And there's quite a number of treatments out there that investigators have used over the years. Copper sulfate is one, tends to wash off quite readily, unfortunately. Ink-based reagents that aren't particularly effective against some of these other tissue elements. And this dreadful sodium borohydride, it bubbles, it's horrible, it's got a short half-life and does absolutely nothing to quench red blood cell autofluorescence. What we've done is we have developed a product called TrueView. And again, this is an adjacent section, believe it or not. You can see essentially zero staining on this adjacent section when treated with TrueView. It drops out all of the autofluorescence from red blood cells, your collagens, and aldehyde fixation. There is another source of autofluorescence, I'll say, that investigators face due to something called lipofuscin. Lipofuscin is really an accumulation of proteins, lipids, etc., in predominantly CNS aged tissue, brain, spinal cord, things like that. And it has more of a punctate type appearance than what you see with the tissue elements I just showed before. Now, I pulled this particular image from the reference I've indicated below here. These investigators did report a double false positive due to the presence of lipofuscin, as indicated by these arrows here. This, look at this 5-HT or MOR1, but unfortunately, it seems to be that lipofuscin was contributing to the interpretation of their results. So what do you do with Lipofuscin? Certainly, I would recommend looking at some of the ink-based reagents that are available out there. And I will say that I did take this image directly from the Biotium website. They showcased that the ink-based reagents, something like Sudan Black, for example, is pretty good in the green and the red channel for reducing this sort of punctate Lipofuscin staining, but it does tend to increase fluorescence in that far red area. But they do have a product that seems to be effective across the channels. So indeed, if you're faced with autofluorescence in your given prep, and this again would be, you don't put any detection reagents on, you simply just look at the preparation itself. Is there any autofluorescence? Identify the source it's coming from. I've listed them here. We went through the tissue elements or due to aldehyde fixation or Lipofuscin, and then employ the correct methodology to try and quench that. Okay, let's work now on absence of staining. And as I've indicated in this image here, this is a pretty big problem for some folks. Maybe, for example, they've been using a given application for a while, working great last week. All of a sudden, they come in on Monday morning, apply their reagents, do their staining, and they've got nothing. This can be quite frustrating for investigators, and like the cartoon I've indicated here, you're left scratching your head thinking, what's going on. Some folks can arbitrarily just go out, they buy new reagents, but they end up with the same result. I would say in most cases of absence of staining, it's probably due to something such as an omission of a reagent, possibly the wrong primary antibody, or maybe a misstep in the workflow, such as antigen unmasking was inappropriate, or indeed the incorrect secondary antibody, or indeed for the case of immunohistochemistry, maybe an incorrect substrate was applied for the enzyme that you're using. But so you're not going to end up scratching your head like this. What you should do to alleviate the cost and drama and headaches associated with this when faced with this particular scenario is to run some defined positive and negative controls. Now, we've just gone through deletion controls in troubleshooting the background, but what we're going to look at here is the reasons why you'd want to run a positive control and a negative control. Now, positive controls are associated mostly with assay sensitivity and specificity. Is everything working as I expect it to? And then the negative controls to identify false positive staining that could be arising from any part of the detection system, actually. Okay, so at this point, though, I'll just pause for a moment and just say, I do field inquiries from investigators all too frequently that apply these positive and negative controls inappropriately or infrequently. I will say that. Now, the idea of the controls, they're complementary to your test specimen. If you don't run these controls, any staining you get on your test specimen is essentially meaningless. It really is. So this is why you have to put in these known positive and negative control sections. They're supposed to be used synergistically with your test specimen. Okay, so to ask the question then, Craig, what is a positive control section? And I've got a bit of a definition here. It's known to contain or express a moderate to a high level of a target antigen, preferably the target antigen you're looking for. So the example I've got here is a piece of tonsil stained here with DOB, hence it's brown. It's looking at this CD20 marker in tonsil. It's a B cell marker and gives you a nice robust stain. And as a positive control indicates here, it incorporates the entire detection methodology. Now, I've also highlighted here Ki67 or Ki67. It's a mitotic indicator. You can also use that on tonsil as well. And I'm really just showcasing here that you can use the same tissue for multiple different markers. Indeed, you could even use tonsil, depending on the cut you've got there, to look at epithelium staining as well, or a cytokeratin marker. So this might alleviate concerns for investigators that are indeed worried about costs or trying to source positive control material. So you can use the same tissue block for multiple different markers. Some considerations when using a positive control, though: sections do not have to be from the same organ or tissue type, or indeed even the same species as your test specimen. However, if indeed you were going to use a different specimen or species, I should say, just make sure that the primary antibody that you're using on your positive control also recognizes the material in the test specimen. And that could be an unknown for you. This is another reason why you'd want to run a positive control, to alleviate, absence of staining. Where did I go wrong? Did it work on my positive control? And then you can showcase it running in parallel with your test or your unknown specimen, and verify whether or not the staining is accurate, or whether there was an omission possibly in your workflow. Now, sections do have to be treated, this is very important, do have to be treated in the same manner as the test specimen. And what I mean by that is, for example, if your specimen was formalin fixed and paraffin embedded, then the positive control material also has to be formalin fixed and paraffin embedded. And certainly if it was acetone fixed and frozen, then the positive control has to be acetone fixed and frozen as well. I've got a little note down here about double antigen staining or multiplexing, for example. What do you do in those cases? I would highly recommend breaking down the particular assay. You will have to run positive controls for each of the target antigens you're looking for to make sure that you are getting appropriate sensitivity and specificity as per that multiplex. Okay, let's move on quickly to negative control sections. Obviously, a negative control is a section that's intended to show an absence of staining. And I've sort of shown that over here with this stain here, just showing a blue hematoxylin nuclear stain. But the reason for employing or implementing a negative control in your assay is to ensure the detection reagents, which include the primary antibody, are not generating any false positive staining that you're going to then report as being where your specific target antigen is being expressed. Now, there might be some end users out there who are saying, you know what, we have this covered. We simply omit the primary antibody, and others have published on this, so we're good to go. And I've showcased that, yeah, I'm aware of that. Certainly in these two publications, JBC, Osteoarthritis and Cartilage, the authors of these papers clearly indicated that they simply omitted the primary antibody as their negative control. For me, I don't find that as being a publication-worthy negative control. It doesn't really address whether or not the primary antibody is contributing to any of the staining. Maybe it's being bound inadvertently by something in the tissue prep. And this is where issues about reproducibility come into question, people trying to duplicate studies. And quite frankly, then, I think that there's an opportunity, a teachable moment here, that we can maybe strive for doing some better science, and simply omitting the primary antibody to me is not really what I'd find as an acceptable negative control. Okay, so a quick question then, Craig. What is an appropriate negative control? And for most end users out there, I'd say substituting a primary antibody for non-immune IG would be the way to go. An example, if your primary antibody is a mouse IgG, substitute in a non-immune mouse IgG at the same concentration under the same conditions as your primary antibody. Now, this non-immune IgG might be a pool of mouse IgG 1, 2A, 2B, etc. So if you do start to see some background with this particular negative control, have a look at the primary antibody. Maybe it's an IgG 1, 2A, 2B, etc. Then substitute in a non-immune mouse IG isotype to match that primary antibody. And that drops out, of course, all the IG1s, etc., 2Bs out of that pool and maybe give you a cleaner result. All right, so let's close out the webinar here with a bit of a discussion on inconsistent staining. And what do I mean by that? I don't mean that your staining works on a Monday and not on a Tuesday. You know, if that's the case, then please go back and revisit what we just mentioned about absence of staining and the need for these positive controls in place. But what I'm referring to here is a patchy localization of the target antigen on the same specimen in the same assay. So imagine these two particular images I've got here. Run in the same assay on different slides. You're getting some nice defined nuclear staining here. You can clearly see it contrasted with this blue hematoxylin counter stain. But maybe 3 slides down, you're starting to see this, and you're starting to see this irregular or differential staining intensity between slides in the same assay. And this is a little confounding to investigators. But importantly, one thing I will say is that don't blame the detection reagents. It's not like the detection reagents are saying, I'm just going to stay in this section and not this section. They don't do that. So the detection reagents are obviously working here, everything from primary antibody through to detection reagents, the enzyme substrate. And so you're going to have to look further afield. But one thing I'll point out, this heterogeneous staining pattern is quite normal for immunofluorescence, immunohistochemistry. So don't try and solve for this problem within a section, solve for this particular difference that I'm indicating here. All right, so what are the things that cause inconsistent staining? I've listed here 4 bullet points: improperly fixed, primarily seen through immersed fixed material. Maybe the outer part of the tissue is nicely fixed, but the more you cut into the block, maybe it's not as well fixed, and that will start to generate inconsistent staining. So for larger, thicker blocks of tissue, make sure it's fixed an appropriate period of time. Incomplete paraffin removal, that could be either due to use of your xylenes, your toluenes, or whatever solution you're using. Make sure they're fresh and that those sections are applied and immersed in that material for an appropriate length of time. Sections can dry out during the application, which would cause inconsistent staining, not just on a section, but within a section as well. And this does tend to arise if you're running a lot of slides at the one time. If that's the case, you may want to break up, do it in smaller batches to alleviate that drying out. And then just make sure that the reagents themselves are dispersed evenly across the specimen, and there's no bubbles present that might be physically preventing the detection reagents from interacting with the tissue itself. But I will say the largest hurdle investigators come across with inconsistent staining, or the biggest reason for that, at least in my experience, is the antigen unmasking methodology. And what I refer to this is largely high temperature antigen unmasking or retrieval. They might be using it it doesn't really matter, pressure cooker, a vegetable steamer, an autoclave, or indeed a microwave oven with all the different makes, models, and wattages. 4 things you got to control here, though. Certainly the time, the temperature, the pH, and the salt solution you're using. Maybe I didn't say that in the correct order. Maybe you need to establish the salt solution, make sure it's the correct pH, the material is immersed for the appropriate time and at the appropriate temperature. And essentially what you have to do, and this particular topic is almost a webinar in itself, and I really can't do it justice. Actually, I've run over time here. So I'm going to just finish off by saying you have to establish the parameters of your antigen unmasking on your positive control sections for each antibody that you're working with. So at this time, I'd like to say thank you very much and open the floor up to any questions you may have.
Great. Thank you for that presentation, Dr. Powell. For the audience, don't forget to submit your questions to the instructor to the Q&A tab on the right side of your screen. We already have several questions to address, so let's get to them. Craig, the first question is comparing IHC and IF. Is one application more sensitive than the other in general or in perhaps particular situations?
The comparison there is between two different applications, immunofluorescence and immunohistochemistry. While we did show similarities in the workflow in the webinar there, a series of sequential incubations, they're really a little bit like apples and oranges, whereby I have had investigators call me up and say, look, I've been getting some great results with immunohistochemistry, whereby they might be using, say, a two-step methodology with an enzyme substrate, getting some nice staining there. But maybe they go to an immunofluorescent application, whereby they're using a secondary antibody conjugated with a fluorophore, and either they're seeing trace, weak, or zero staining, and then they're saying what's going on. So the sensitivity here, that there are two different applications. The two-step methodology with the enzyme-based, in that example I just gave, would be significantly more sensitive. I'll throw a dart at the wall and say maybe 20 times more sensitive than the fluorophore conjugated secondary antibody approach. So investigators would have to be cognizant of that level of amplification and sensitivity. So it's very difficult to draw a straight line between those two. I will say that there are advantages of one over the other. In some cases, certainly for immunofluorescence, can give you very fine, exquisite detail, depending on what you're looking for. Intracellular localization is fantastic for immunofluorescence, and certainly immunofluorescence does enable you to do co-localization detection of reagents as well. One on top of the other, co-expressed in the same cell compartment of the same cell. So certainly there's some pros and cons to both of those, but drawing a comparison between sensitivity a little tough. You can certainly employ other techniques in fluorescence to increase that sensitivity, to make it comparable to immunohistochemistry, absolutely. But just with that example I gave, just be aware that it is a little difficult to make that comparison.
Great, thank you. When it comes to detection, why might someone choose an alkaline phosphatase IHC detection method over HRP?
Yeah, certainly I would say that the use of peroxidase-based methodologies and the use of, say, a substrate like as DAB, diaminobenzidine, probably the most prominently used methodology out there. It does give a very nice, discrete deposition of color out there on a single label prep. But let's just say, as I mentioned before, earlier on there, I was talking about some quenching for endogenous peroxidase activity. Some investigators who might be working with highly vascularized material might just be encountering just a really lot of stubborn, I'll use that phrase, in peroxidase activity that's very tough to quench. There were a couple of publications there. One of them uses a glucose oxidase, sodium azide, I think, methodology. It is very effective, but for those of it is a longer, more involved process. But swapping to alkaline phosphatase would circumvent having to deal with this persistent peroxidase activity. That's number one reason. I think another reason would be it gives you a different choice of color with some of the substrates as well. You could improve the contrast with potentially inherent pigmentation or melanoma or whatever you've got with some of the different substrate colors with alkaline phosphatase. Or indeed, if you're doing double labeling or triple labeling, you can look at different colors as well. And some of the alkaline phosphatase fluorophores do fluoresce. So that's another reason why you may also want to choose that over peroxidase.
Great, thanks. We received several questions about the TrueView reagent. First, where in the protocol for IHC is the best place to use it? And does it survive organic solvent treatment?
Okay, so number one, TrueView is actually applied in immunofluorescence.
Oh, right, sorry.
No, yeah, no worries. Just to clarify there, so it's actually the way it's usually set up is it's done after you apply your fluorophore conjugated detection reagent. So let's just go back to the workflow example I used. You might have an unconjugated primary antibody. You put on your fluorophore conjugated secondary antibody, let that incubate for, say, 30 minutes at room temperature. You do your buffer wash to remove excess. Then you'd apply the TruView reagent for about 2 to 5 minutes. Then you do a PBS wash, and then you'd apply your anti-fade mounting media. So that's hopefully gives you a bit of an idea of where in the workflow it would be applied. And how quickly, or it's quite effective in terms of its use. It only takes about 2 to 5 minutes to reduce that autofluorescence. Is it stable through the use of organic solvents? And that's a good question. I think that we've only used it with aqueous phases. And then like aqueously based mounting media or anti-fade media, such as Vector Shield or things like that. And so I don't know that I have an answer whether or not it would be suitable for use, say, in a permanent mounting application where you might then go through xylenes or toluenes or things like that. I don't know that that's a parameter we've actually evaluated.
Okay, and what tissue thicknesses does it work with?
Yep. So most of the work that we've done at Vector Labs would be on tissue sections 10 microns and less in thickness. So we do know it's quite effective there. For thicker preps, if you go up to say 20 microns or a little more than that, certainly I would recommend applying it for a longer period than what we recommend. Instead of the 2 to 5 minutes, you can leave it on for 10 to 20. It's not that it's going to be problematic or detrimental to the fluorophore or anything you've got there. But that could be helpful. I did encounter one investigator. I think they were I think it was New York. They did show me a 200 micron section that they used with TruView, but I don't have the parameters on what that particular investigator used. But she did send me a picture, and it was very nice. It was a 3D image there, and she did say that she used TruView on that, but I don't have a procedure on that. But nonetheless, 10 microns or less would work very well.
Okay, great. Is it better to wash for fewer times for longer periods of time, or more frequently for shorter periods of time?
Yeah, how long's a piece of string on that one? That's a good question. But I would say I don't know there's a definitive answer there. It would be whatever is effective to help reduce toning background, if you do get that, or indeed to remove the unbound material from your tissue prep. And just to segue back a little bit to the inquiry that some investigators might be using some thicker preps, usually a little longer if you are using thicker preps does work better than shorter applications. So try and see which ways work best. But certainly for, say, 10 micron sections or less, and we usually work with, say, 4 to 6 micron sort of sections, we usually, for both immunohistochemistry and immunofluorescence, usually run, 2 to 3 minutes, 2 to 5 minute type buffer washes in between incubations. And if you were going to go with thicker preps, 3 to 5 changes of 10 to 20 minutes is not unheard of. And certainly this might be for, say, embryos or xenopus, I should say, or zebrafish or something like that, if you're working with those sort of preps, or indeed, thicker material.
Great. And we have time for just one more question. Can you elaborate on what other negative controls would be appropriate beyond substituting non-immune IgG?
Right. So in the webinar, I did mention about the use of substituting a non-immune IgG as a negative control. Probably the next best negative control for most investigators would be applying an irrelevant antibody. And what I mean by that is an irrelevant primary antibody, one that will not bind to anything in your given preparation. Let me give you an example. So let's just say you're working on CNS tissue, on brain tissue. You're pretty sure that there's not going to be any, stratified keratinized epithelium in there. So if you're working on NUN or GFAP or something like that in brain tissue, use a primary antibody raised in the same species of the same isotype at the same concentration against that stratified keratinized epithelium, and then use the detection reagents after that. The idea being is to show that primary antibody isotype from that species is not being bound inadvertently by some tissue component in there and contributing to staining. So that would be the easiest, more straightforward negative control. There are others, depending on the study they've got. If you've got a knockout model, that'd be great. An absence of the antigen, maybe there might be a possibility to remove the antigen, either through enzyme activity, maybe through digestion. And then there is one other control that might not be practical for a lot of investigators, would be to incubate the primary antibody with the immunogen used to generate the antibody. And a couple of things there. That might not be available, as I mentioned, if you're getting the antibody from a commercial vendor. And number two, the question's been raised whether or not that antibody, while it does bind the immunogen in solution, is it a true representation of what that antibody is binding on the tissue prep, particularly heterogeneous tissue prep? So I think the first negative control I mentioned, the irrelevant primary antibody, would probably be the best way to go.
Great. Thank you for that in-depth discussion, Craig. Unfortunately, that's all the time we have for today. For further discussion, feel free to reach out to the instructor directly. Their contact information is on the screen. Make sure to follow the scientist on social media for updates on future technique talks. Our thanks to all who took the time to join us today, and special thanks to our instructor for today's course, Dr. Craig Powell, as well as our sponsor, Vector Laboratories. Have a great day, everyone. Goodbye.
Thank you.